talk today. So we're getting back to some immunodeficiencies, things that actually relate to blood cells. So this is very apt for the Department of Hematology. I know there are some in the audience online from there now, as well as the Centre for Blood Research here. And it's a great pleasure to introduce to you Dr. Stuart Turvey, who's at the Children's Hospital here. He's a pediatric oncologist. So as soon as he opens his mouth, you'll see that he has a cultured accent, just like mine, from Australia. And he was born in Sydney, like me, actually. And then he went to med school, unlike me, and in Australia for 6 years, and then he became a Rhodes Scholar. And for those in the Commonwealth, which we are here, this is nothing to sneeze at. And so he went to Oxford then and did a PhD in transplantation immunology out of the Headington Hospital. So at this time, he hadn't practiced as a doctor at this point. So then he went then to Boston and for 8 years. So he then did his residency in pediatrics. And that was followed by his fellowship, a combined fellowship for 3 years in allergy, immunology, and rheumatology. And so he became more of an immunologist at this point. So he's had an interest in immunology and in transplant immunology, so bone marrow is being swapped out or stem cells being swapped out. And now here he's, I don't know if are you ahead or something of the, just riff-raff here? Yeah. So he's a pediatric immunologist and involved in immunodeficiencies. And this is where I came across Stuart. He gave me a call one day, and I hadn't heard of him, but he had a patient that had a MALT1 deficiency, and some of you would have known and heard these stories. And MALT1 is a protease. It's involved in NF-kappa B activation, but he had the only living MALT1 deficiency patient in the world. And I don't think you're talking about that story today. Yeah, but the great thing about that, it really exemplifies the strength of the Canadian health system. I mean, people poo-hoo it, and the Americans poo-hoo it and stuff, but this kid came from a family of immigrants, or refugees rather, from Kurdistan, which was a Kurd, probably from, from Turkey or from Syria? Yeah, Iraq. And came out at that time. And so cousins were coming down. And so she was a double recessive for this MALT1 deficiency. But she got full exon sequencing as an immigrant, as a refugee, and then plugged into a system of excellent healthcare. At 14, she weighed 20 kilo, right? Incredible, like that she's still alive, teeny tiny. And then in my lab and with Stuart and then with Mike's lab, we all three got together, and then with Novartis, we ended up developing a molecular corrective drug for her mutation. It wasn't used, it could have been used. It would have been safe and effective and efficacious, but she actually had a transplant at that point. So that's how I've come to know Stuart, and he's great. And you're going to hear some really great stories. Yeah, because I don't invite riff-raff here, so so you're going to hear some really nice stories today, and it's a real pleasure to welcome you, Stuart. Thank you for coming.
I think it's too wireless. Can you check it?
Low. We don't need it. Let's just use this one. Okay, I think we're good. We'll talk to this one.
Yes.
So thank you for that kind introduction. It's a huge honor to be here. I'm a little nervous, I have to say, because I'm going to talk. This can be a more clinical talk, maybe than traditional for this audience. But there's a reason for that, and it's really, I think Chris alluded to it. I think my passion that we can link the patients that we see through our universal healthcare system to the basic science capacity at this, in world-class university. And so there's a few little examples of that, but I guess I'm sort of here as also offering to be the bridge, if I can. So if there's genes you're really interested in, or patients at our children's hospital, we may well have patients who have a defect in that gene or so on. So that's my offer. Let me get myself organized here. So this is me, this is what I do, right? I'm a pediatrician. And so the research we do really starts from this interaction. It starts with the interaction with the patients. And then, some of those will have unusual and novel conditions that we'll take into the lab to try and understand what's going on. And this might sound like an oxymoron, but for us in pediatrics, rare diseases are really very common. And so the numbers are quite striking. It's estimated that about 1 in 3 children admitted to a pediatric hospital, like BC Children's Hospital, actually has a genetic, do something here. Oh, no, I better not push the button. Has a genetic disorder. We're making nice progress, but there's still huge numbers of these disorders that don't have a name yet. And so patients who are born with rare diseases without a diagnosis undergo this diagnostic odyssey of test after painful invasive test, futile specialist visit after visit. And with access to next generation sequencing technology, we've been able to end that diagnostic odyssey for more and more patients. And I think why that's so important is that we also have a growing armamentarium of medicines. And so we're really able to start to practice precision medicine, where we can precisely target and treat that patient's condition. So this is the sort of era that I feel that we live in now, and it's exciting era. And for me, the massive transformational and disruptive change in clinical medicine that we're undergoing now is this. And that's the price of sequencing a human genome. And so I started my faculty job in 2004, and the genome was inaccessible at those times, tens of millions of dollars to sequence a genome. This data suggests around $1,000. There's a new company called Ultima that's bringing a new short-read sequencing technology that's suggesting $100 they'll sequence the genome for. And so I just don't think anything has changed in medicine as much as this. And so now we're able to sequence easily. We're able to use it for molecular diagnosis for infections and other things. And the problem, well, maybe I shouldn't preach to this audience, but the clinical problem is that the people who kind of are the mentors, the people who set the exams and write the textbooks, grew up in the inaccessible genome era, and our residents and our patients live in the very accessible genome era. And there's a massive gap actually in understanding. And we have to fix this. So this is a hematology thoughtful group. The analogy might be with a blood count. So pediatricians do blood counts. We see a child's anemic. We feel comfortable, for example, to identify iron deficiency and start iron supplementation. The flip side is if a genetic test comes back, no one feels they can understand it, and they all want to run to the medical genetics doctors, and they're overwhelmed and can't do this. So we need to, this price point just makes this accessible, and so genomics becomes the job of, I think, every subspecialist and maybe even every family doctor in the near future. And so when we think about diagnosing Mendelian or single gene defects, we can see this massive transformation over time with the introduction of next generation sequencing or whole exome sequencing. The first clinical application was only in 2010. So it's really remarkable. And now that space, is dominated, gene discovery is dominated by next generation sequencing technology. And so for me as a clinical immunologist, these are exciting times because the number of diseases we know about has accelerated profoundly. So again, if we go back to 2004, started as a proud faculty member, there was about 100 diseases. I think I probably knew most of them. They were classic immune system disorders. absent B cells in, X-linked XLA, or absent T cells in boys born with severe combined immune deficiency. Now the number has accelerated along with the access to sequencing. What's different about clinical immunology discovery space actually is the fact that about 25% of these patients were initially, or diseases were initially described in a single patient. And that's unusual in genetic discovery space. Typically, genetic discovery involves building cohorts of patients who have similar genetic change, similar phenotype. But the advantage we as immunologists, hematologists, blood doctors is that we can access the organ relatively easily, and we can interrogate primary cells of patients, and so really prove causation in a more powerful way. And you could contrast that, for example, to neurodevelopmental challenges where it's hard to access the organ. I guess we can grow iPS cells and things now to model it, but it's been less accessible. And this is the 2025 update. So this is exciting times in immunology when they update the list and tell us how many. And so just about a week ago, we added 63 new disorders in that last 2 years. And if I brag a little bit, we contributed about 10% of that global knowledge from patients who came through our universal healthcare system, funneled down across BC and the Yukon, and ended up with us. So I'm excited about this because I think there's huge discovery space for us to make those discoveries from our patients. And then I want to sort of bring it back to the campus here and for basic science discovery to really understand the mechanisms at play.
What about sick kids in UT?
Yeah, we're better than them.
Yeah.
So this is taped, but yeah, we have done. I mean, so yeah, exactly. So the Sick Kids Hospital is a very good hospital and has very proud tradition of leading this space. And we had, but we've caught up. Yeah, they're good. I don't want to be a jerk, but we're pretty good too. And so what we're talking about are called inborn errors of immunity. That's the favored term now. Might have been previously called primary immune deficiencies. So genetically determined disorders of the immune system, greatly enhancing susceptibility to infections, but also other forms of immune dysregulation. When we add up those 500 to 600 diseases, the cumulative prevalence is actually pretty large. As 1 in 1,000, 1 in 500 are some of the estimates. And so when I talk about this to my clinical friends, I remind them that, if you're an obstetrician, you'll have 500 or 1,000 patients on your roll if you're a psychologist on your roster. So these patients are out there, often not recognized, to be honest. That early diagnosis can be life-saving. We can offer very accurate genetic counseling for families about future pregnancies or family members' risk. And then I think what's also really powerful and maybe most relevant for this audience is that these diseases provide fundamental insights into human biology. And we've advanced hugely, for example, with genetically manipulated mice, knockout mice and others. But now, truthfully, we're able to find the human equivalents of those. The irony is when I write my CHR grant, they say it's just one patient. And, how do you know it? And I want to say, well, your mouse is just one mouse. You just had lots of babies of it. But so not to pit these discovery streams against each other, but just to say that I think it's exciting to be able to match that biology. And the other big change is our tools that can interrogate multi-parameter flow cytometry or proteomics and other things in small samples. So we can really start to do a lot of what we did in mouse models in humans, in primary samples. And so Chris alluded to this. I sort of talked about this advancing human understanding of human biology, which is like a huge passion for me. And, this is just sort of showing a handful of those papers. But yeah, I came to Chris and said, I have this girl with MALT1 deficiency. I bet you can use your fancy tools to find what MALT1 should cut in her or doesn't cut in her. And that ended up being, I think, a really powerful discovery stream with that has also been really fun. So, it's a win-win for sure. Just put that one in twice. And so when we as clinical immunologists sort of think back to our history, most of the emphasis has been on infection. So on the right, I'm showing you David Vedder, the bubble boy, born in the 1970s. Interestingly, his older brother died of infection and was worked out to have no T cells. He had a severe combined immune deficiency. David came along, they put him in the bubble, and he was protected from infections. The sort of sad part of that story, maybe not surprisingly, is this was terrible for his social and emotional and neurological development. And as a teenager, he was given a bone marrow transplant and died of overwhelming EBV quite quickly after that. And there's health charities here I'm showing you on the right called the Jeffrey Modell Foundation. It's a remarkable charity that's been raising awareness directly to patients. They said, we'll skip the doctors. We can't teach them things. We'll go straight to the patients. If you have these warning signs, you should be checked for immune defect. But again, all those signs talk about infections. And what I've been emphasizing really is that those infections are the tip of the iceberg. And when an immune system doesn't function well, the patients will have malignancy, autoimmunity, or allergy. And the allergy bit has been the least studied, to be honest, and that's an area where we've made some nice progress. And so I wanted to sort of share a story about single gene defects causing allergic disease. And in recognition of the importance of this group, John Lyons and Josh Milner termed this coined primary atopic disorder to describe single gene defects that cause allergies. And they're really a subset of that larger 500 or so. And there's about 50 disorders recognized. And as we look through those disorders, we see that they reflect many different disease mechanisms. Some are disruption of the skin barrier, allowing the allergens to get in. Others are failure of immune tolerance, for example, the absence of regulatory T cells. I'll show you a story about altered cytokine signaling. So we can start to understand them mechanistically, and that informs our clinical treatment. Bear with me for like one more sort of clinical part of this. But so the challenge I think clinicians have, we have is, how do we recognize the rare patient with that single gene defect in the context of allergic disease? So common polygenic allergic disease affects up to 30% of the population, and clearly they don't all have single gene defects. And so we've done a lot of work in that space, trying to generate clinical red flags that might suggest a patient should be checked for one of these single gene defects. And to summarize all of that, it really means that they have the allergies. You sort of got to have that to get started, but then they have other things that aren't typically a feature of common polygenic disease. They develop autoimmunity, they develop malignancy, they don't grow well, which is sort of a big integrator of health in pediatrics. They may have infections. And then interestingly, they often have these connective tissue disorders, easy fractures, abnormal face structure, lung damage, and pneumatocysts. So this is the kind of group that we've been looking at. And so with that background, I'll take you on a journey now. It's sort of translational journey that we've been on, looking to describe a new human disease, and also sort of excited to get input on maybe how we can really define and probe the molecular mechanisms at a more sophisticated level. So everything in my life in the hospital usually starts in the Starbucks lineup. Although like the app has sort of messed up that relationship. But, no question. Okay. And so I was in the lineup and met with someone you might know. He's retired, just retired. His name is Mike Sear. So he was a pediatric lung doctor who'd worked at our hospital for 30 years. He was the guy who saw vast numbers of patients and remembered them all. There's like some of these people. I remember their names, I remember everything. And he said, you know, Turvey, you study these rare diseases, you got to go find this guy. He's probably 30 now, and he's going to have one of your diseases. And so preaching, doing what I preached, which was practicing what I preached, we went and tracked this young man down. And this is his story, and I sort of share this in the classic clinical grand round style. So he was the first and only child of Caucasian Canadian parents, lived sort of, you know, in the lower mainland area. The family had no real health problems. And then he was born, looked perfect, but within a few weeks of life, started developing widespread, very difficult to treat eczema or atopic dermatitis. And so babies often get sort of eczema on their cheeks or in the creases of the knees or the elbows. This was all over and was just this struggle for his whole life to deal with. And then as time went on, he developed more and more allergic disease. And so he ended up with multiple IgE-mediated food allergies. He couldn't really swallow and would vomit a lot. And the gastroenterologist identified that he had something we call eosinophilic gastrointestinal disease. So the eosinophils in the esophagus and throughout reacting to these food allergens. He had asthma, failure to thrive. He didn't have so many infections. We immunologists get excited about infections. These were kind of infections that you do if you disrupt the barrier by scratching so much. They weren't really deep-seated infections. Over those 30 years, at the diligent doctors at BC Children's Hospital gave him all the best treatment, and nothing controlled his symptoms other than ongoing doses of prednisone, so high-dose corticosteroids, with all the side effects. He had high eosinophil counts in his blood, high IgE, and so we called him. He participated in the research project. We sequenced. We found a genetic change that we thought was the answer. We called him back. That takes us 9-10 months sort of thing. We called him back, and the family said, he died last week, and he died of a ruptured cerebral aneurysm, so died of hemorrhage. And in fact, this is not chance, and I'll show you that, but the genetic change in this is also important for developing the vasculature.
I'll move over there after this.
There's someone who's making a bit of noise.
I just need to set up the phone. Yeah.
Dr. Moon, can you turn off your, go on mute, please?
Thank you.
Oh, they might have, it's good. Okay, well, let's get going. And so pediatricians love growth charts. We track growth. It's a great integrator. And you can see these little red dots on the height and weight, and you see that he's below the 3rd percentile. So those lines are the 3rd percentile. So that is how we define failure to thrive. So his height and weight below the 3rd percentile. And so this is how we handle these types of conundrums, really, is clinicians will come to us, or we'll see them in our clinic. We'll go ahead and do next generation sequencing, and that has changed over time. Right now, we typically would do trio, so the parents and the child, short read whole genome sequencing. That's sort of effective, cost-effective point. Using bioinformatics strategies, we'll filter the list to candidate genes that seem to match the phenotype. We'll do experiments in the lab to prove that the genetic change is damaging. We'll test treatments to see if we can normalize that. And then sort of the big part that's sort of defines the global clinical immunology community is actually one of collaboration. And so interestingly, for these things, we often then just write to the world and say, hey, we have this new disease. Does anyone have one who looks a little similar and want to join us? So that's a sort of interesting, risky strategy because you maybe get scooped. Well, how do you do it? So this Clinical Immunology Society has a listserv that really is like global. And so I write on it, hey, do you have any patients like that? There's this thing called Gene Matcher, which is like dating for, like internet dating for genes. So you write your gene, and you put your email in, and if someone else writes the same gene and puts their email in, you get an email and say, hey, you might be friends. And then it sort of starts a little coy. You say, oh, my patient has an immune system problem. And they say, oh, mine has a brain problem. And then you don't work out. But if they have an immune system problem, then you start talking. And then we also used yeah, exactly. And we also use preprint servers, you know, like put it up there, which is also the least I can say I was first in the public domain if someone else scoops us. But this is what we did and that process. And so through that, we recently identified a condition caused by heterozygous germline gain of function genetic changes in the gene STAT6 as the cause. And so that's the cause for this young man. And I'll just sort of tell you how we get there, and then again, would really appreciate advice on how to do better. So this is the science bit now. So STAT6 is just a no-brainer genetic change for allergies. So STAT6 is a transcription factor that sits at the heart of allergic inflammation. And so in my simplistic mind, the key pro-allergic cytokines are IL-4 and IL-13 that signal through their receptor, activate the JAKs, which then go on and phosphorylate the STAT6 then moves to the nucleus, drives transcription of pro-allergic genes, including more IL-4 and IL-13, and we have this pro-allergic cycle. Was that okay, Kelly? Was that a fair description of? And and these molecules kind of sit at the heart of what we see clinically on allergies. So whether it's B cell class switching to make IgE that then mediates that type 1 hypersensitivity reaction on re-exposure, or where it's changing the lungs, the airway remodeling with the fibrosis, the proliferation of the smooth muscles, the mucus, all that biology that underlies asthma helps recruit eosinophils. So it's a kind of no-brainer target for being related to allergic disease. The other interesting thing from us as immunologists is that this JAK-STAT pathway has been enriched over time with genetic changes that cause primary atopic disorders. The classic one that people might remember is Job syndrome. It's caused by dominant interfering changes in STAT3, named after the biblical figure Job, who had boils and other problems. So that's sort of the most famous, I would say, primary atopic disorder. STAT5B is also another disorder. And then increased activity of JAK1 is a condition that we described actually a few years ago. So we've been filling in that space. So the other way to sort of do human genetics is, rather than focusing on very rare diseases, is to do population genetics, taking larger groups and doing genome-wide associations to ask the question, is genetic change within that genomic region associated with the phenotype? And so STAT6 had come up for more than a decade in tens of genome-wide association studies as being linked to a large number of allergic phenotypes. So it was a very hot candidate to make good sense biologically, but no single gene defects had been described. And so I did this, dating process I described, finding friends, and like really quickly, like within maybe 3 or 4 months, we moved from this one patient in BC to a consortium that spanned here Vancouver to Hong Kong. And so we moved really quickly, and subsequently many more patients have been found with this, which I think also emphasizes that it's not so rare and was just ready to be found. Okay, we're doing a journey through different things. Now we'll do some human genetics. And so these are the different families we have. And if you've got sharp eyes, I point out the bottom right, family J. So family J has 3 affected generations. So they're colored in black. And then if you look, you'll see that they're different sexes. Females in circles, males in squares. So that's really consistent with an autosomal dominant genetic inheritance pattern. And you can see that coming through with other ones, family C and family F. I think the striking thing, however, is that more than half of these families, it was sporadic disease. And when we sequenced the families, we found de novo genetic changes in STAT6. So not inherited from the families, happening de novo. And that's a theme in issues of human and clinical genetics as we've sequenced more and more patients, is that de novo disease is much more common than we would have predicted. And so when I went to medical school, they told me, ask a history, and if people have it, that's a genetic disease. And that's obviously still true, but the flip side is it may still be genetic if it's a sporadic condition. So that's, I think, really striking and really emphasized in this. And the patients all looked basically the same as that young man I described, with the multi-system allergic disease that began very early in life. And I'll show you a little bit about how it looks, just to get a sense. So this is the boy from Hong Kong. This is his skin. So this is very bad, thick, difficult, sort of impossible to manage allergic disease. He had great clinicians in Hong Kong. This was as good as they could get his skin. This one's a little more obscure, but this eosinophilic disease, this is the looking down with the endoscope at the esophagus there on the left. And there are features, the furrows and then those parallel lines that are actually very indicative of eosinophilic esophageal disease. And the biopsies were really enriched with lots of eosinophilic infiltration. I'll whip through some of this. But also the patients had super high levels of IgE and super high levels of eosinophils in their blood count. And so the gray is the normal range, and this is a log scale. So for us who see, like from the clinical side, we see these, it's really striking. This phenotype would get our attention for sure. And then I often get this question, well, what about all the other things that immunologists like to do? We measure T cells and B cells and NK cells and antibodies and all of that. Those things told us nothing. And so the kind of message, I think, in this discovery space for us, or for the clinical space, is that we should move quickly to genetic testing for patients who are outliers like these patients. That'll get us the diagnosis quickly, and we've been advocating a genetics first approach. It's hard to do that because the system wants us to do kind of old-fashioned tests and then do genetics at the end. But if it's this cheap, this is the way to go. Okay. So now I want to talk about some of the genetic changes we found in these individual patients. So we found heterozygous genetic changes, so one copy affected genetic changes in STAT6 in the affected patients. They weren't all exactly the same genetic change, mind you. So we needed to interrogate that. And one of the first steps we like to use is population genetics. So we have now ever-growing databases of normal human genetic variation. And so the biggest one is called NOMAD, and now has close to 1 million people sequenced. So we start to understand normal genetic variation, and so can assign allele frequencies in those larger populations. And what we found for the patient variants here in the red is that none of them were in the population databases. So these were not just, polymorphisms that were out in the population. These were not reported in the healthy populations. And then the other trick we use are bioinformatics tools to predict how damaging. And so these genetic changes were all missense mutations and amino acid changing mutations. And so we can use tools. The one we like the best is called CAD. And we typically see that a threshold above 20 is suggestive or predictive of it being harmful. So from this data, we see that we had vanishingly rare genetic changes that were all predicted to be damaging. So that helps us think about moving forward. This might be more familiar to this audience, but let's look at where they fell in the transcription factor. So at the top, I'm showing you the linear structure of the STAT6 transcription factor. And at the bottom, the 3D rendering with the DNA double helix in the middle as the circle, and then the homodimer of STAT6. And so there's a few things when you look at this. So first is that something is going on at amino acid position 419. We saw a whole bunch of patients who had slightly different amino acid changes at 419. So that was interesting. And then when we look at the 3D structure, even the ones that weren't in the DNA binding domain clustered around that protein DNA interface. So that was, I think, more evidence that these might be functionally relevant. They weren't out in the tails and in other places. Another bioinformatics trick we like to use is to look at another catalog called COSMIC, which is the catalog of somatic mutations in cancer. So again, sequencing cancers, depositing the somatic mutations we see, and then building a picture of what those recurrent ones are. And this is an excellent resource for getting a hint for which genetic changes might be gain of function, given that somatic mutations often give those cancer cells a survival advantage. And so we know that recurrent genetic changes in COSMIC often offer a survival advantage. And so when we looked in COSMIC, interestingly, that the cancer that's linked to STAT6 is B-cell lymphoma. It's not other cancers, it's B-cell lymphoma. And then when we look at COSMIC, so I'm showing you COSMIC on the bottom and our patient on the top, 419 just jumps off the page as this profoundly recurrent STAT6 change in cancer. And then, but if you look carefully, you can see that a number of the other genetic changes that we identified in the patient here on the top turned up as COSMIC recurrent changes. You can see that. So that was, again, more evidence that these were likely gain of function. If you're still conscious, you might wonder, what happens to these patients in terms of their risk for lymphoma? So I'm telling you that these patients have these changes in their germline. And so are they, one step or more steps down the path towards developing malignancy and maybe particularly B-cell lymphoma? And I guess the answer is we don't know. This is a brand new disease, but this little vignette, I think, is telling. So this family the mother is the oldest person in the cohort. So she was in her 40s and now in her 50s. And she had, one of her sons died of anaphylaxis, actually. And she had B-cell lymphoma. So she developed B-cell lymphoma in her 40s, and then it was treated, and then about a decade later came back as a more aggressive form of B-cell lymphoma. So my sense is that that's not chance, and that that we're advocating that these patients should be assessed and be recognized as really a hereditary cancer syndrome, as well as the allergic syndrome that we described.
Stuart, what you know from studying other patients who have lymphomas, adult population?
Yeah, I mean, we know exactly. So we know this exactly. So COSMIC tells us having a somatic, so that 419, having somatic changes at 419, amino acid 419, are profoundly enriched in B-cell lymphomas. So yeah, we're the sort of the opposite. We're now, this is in this woman's germline. And so, yeah, we predicted, I guess, that this would be the case.
But my point is, there's lots of patients who have lymphomas, adults,
yeah,
lymphomas, there's oodles of those. You could go back and see.
Yeah. Yeah, that's exactly right. Thank you. Yeah. I think the flip side would be, how many of those B-cell lymphomas also had, terrible allergic disease, and that's going to be a really small number, clearly. So I wanted to show you some of the mechanistic work again.
Tolerance, what happens as a kid is different to as an adult. The intolerance and as an adult.
Yeah, I mean, I think STAT6 signaling is really important for B-cell survival. And so I just think that, that survival advantage for the B cells is why it helps develop B cell lymphoma later on, but also why I think these patients are at risk. It also explains, why they have high IgE and other things. It's just this thermostat is turned on all the time. I look at Kelly here; he sort of knows more about B cells than me. So, yeah, the somatic sense. Yeah, I think so too. So I'll just take you through some of the mechanistic work we've done. But again, I feel a little nervous because I feel like we've only really scratched the surface. But I'll take you through some of the relatively simple things we did. So, not surprisingly, we went to the toolbox and pulled out some old favorites like luciferase assay, using a STAT6 promoter element, and then transfecting in wild type versions, some other population versions, and the patient versions. And so, see if I can point. And so we did that here, reading out STAT6 promoter activity. Here's the wild type, unstimulated, stimulate with IL-4 on the log scale, goes up nicely. Here's another population variant that was rare in that population, amino acid change. Again, looked very normal. And here we see all the patient ones. They're all turned on at baseline. We probably saturated this a little, so there wasn't much room to move up, but it is a log scale. So again, suggesting, I think, consistent with a gain of function change. Similarly, we're able to look at STAT6 phosphoflow to interrogate them. And you can see all those colored ones, the patient ones are way over on the right-hand side. What was interesting, if you have very sharp eyes, is this top one here, which is the patient from Vietnam, didn't move in this assay at all. And that's, I think, because it was right next to the phosphorylation site. And so we were able to do Western blotting to prove the gain of function in that one. I think people can see. Can you guys see that? Oh, you can't see it, but they can see it. So I'm pointing. Those guys can see it, but you guys can't see it. So I don't know who to privilege on their pointing. But anyway, you can see get the message. And we also, nicely are able to go to the patient cells and start to ask the question, well, what do we see in the patient cells outside that model? And so it's kind of this crazy juggling act of FedEx and praying that gets the samples from Iran or wherever else. And they would roll in, and Mehul Sharma was the PhD student, and I'd all be excited, let's see the experiment, and do the experiment. And the max sort of patient readout was always the same as the controls. I'd say, do it again, can't be right. And then we took a step back and said, well, you got to turn off immune system function as well as turn it on. And when we started looking at that, I think some interesting things evolved that the patients really had this slower dephosphorylation phase. So in the patient primary cells read out a little differently to the models, maybe not surprisingly, but if we think that the area under the curve is kind of that biological signal, these patients were having more profound signal. We also do some relatively simple things on the patient cells, trying to look at Th2 signature by flow cytometry, looking at different cells expressing the classic cytokines, IL-13, IL-4, IL-5, and again, they were all higher in the patient cells. So this is, I have to tell you, sort of where we've got at the mechanistic level is, I think, we did all the population stuff and the clinical stuff and the genetics, and then we have, I think, some reasonable function showing that these genetic changes are gain of function, but we've only scratched the surface. So I don't know things like, does these patient versions of STAT6, does it bind more strongly to DNA? Could we do ChIP-seq? Does it bind to different places throughout the genome? Could we do affinity measures? So this is actually sort of the part where I feel like the sort of understanding fundamental transcription factor biology and how these genetic changes change that, I think are really ripe for, you know, fundamental investigation and interrogation. So that's sort of where we're at with this. But instead of sort of digging deep in that piece, we took a more clinical lens and said, it's all well and good to tell the patients they have STAT6 gain of function disease. They're not so excited about that. They want to know what we can do with that information, which is quite reasonable. And so the exciting time we live in is this precision medicine era. And so we as clinicians have this growing armamentarium of medications that will target specific parts of this pathway. And so as we look at that, we were really committed to a kind of a drug repurposing strategy. We wanted to look at approved medicines and see if they could be repurposed to help these rare patients. And some really obvious candidates turned up. So one is dupilumab. So that's a monoclonal antibody, this guy, monoclonal antibody against the alpha chain of the IL-4 receptor. It blocks signaling by both IL-4 and IL-13. So a really nice candidate. It's approved down to kids 6 months of age, I think, for atopic dermatitis. And then also the growing list of JAK inhibitors. So oral medicines that really effectively block the JAKs are also becoming more available. And so obviously these targets we're talking about are all upstream of STAT6, but we're really taking advantage of this feed forward cycle, thinking that if we could block some of the upstream feed forward, we might have some nice beneficial effects. And so we tested, you know, in this assay here, I'm showing you phospho-STAT6 again. We tested these against some of the gain of function variants and showed, not surprisingly, that they're all pretty good at turning off the signal. And so we shared this information with our clinical collaborators, and they, you know, went and made some changes to their patients. So here's the young man from Hong Kong, and you know, you can see his skin. We also can do these scores of how bad the skin is, and the scores went down. His skin got way better, but this is my favorite figure. And so I showed you growth chart before. So this young man had profound failure. Here's his height. The doctors, as a teenager, put him on growth hormone. Nothing happened. Here's his weight. And then we put him on dupilumab at the age of 19, and he grew like 15 centimeters, and he doubled his body weight. He went from 30-something to 60-something kilos. And so I got to meet this guy. So I guess, and he talks about just his life. He used to hide in the dark room, feeling like he looked like, you know, a cartoon character and had no life. And through this dupilumab has been really profoundly changed. So it was good reason to get up. Yeah. Yeah. He was, yeah, I mean, so he had profound like growth failure because of this. And I think a lot of it actually is the terrible, like, whole body, but including GI inflammation. So obviously his growth plates were not fused, and he had that growth potential. Yeah, and we see this a lot in very sick kids. The MALT1 girl, after we gave her a transplant at about 15, she did the same sort of thing, doubled her body weight. She's married now and is doing well. So anyway, yeah.
How hard is she?
Yeah, she's this tall, maybe, but you know, she was this tall before. So anyway,
But about the growth plate, so what's that telling us about STAT6?
I don't think I think this is sick kids end up with growth delay. I would say that's sort of a pediatric truism, and you know, particularly like profound GI disease. So kids with bad inflammatory bowel disease have failure of linear growth. You know, they just don't get tall, and then they fuse their plates later. So if we can do things to them early enough, we can make profound changes.
Also a nutritional defect?
Yeah, I think it's a whole body. I think he has a whole body inflammation problem, and so he had a very restricted diet, couldn't eat much. His gut didn't absorb it very well. he was pretty depressed. He just like his whole body problems. He felt better. He could absorb food. And this is what happened.
That versus life now.
Yeah. I mean, another tool we have for clinical immunology is bone marrow transplant, hematopoietic stem cell transplant, but that's not going to work. It works well for hematopoietic-based disorders, right? STAT6 is expressed in blood cells, but is expressed beyond. So we would say that dupilumab is safe, really effective. This is a good, but it would be a for-life medicine because this is encoded in his germline.
And I would assume that he's got an advantage in cells that have status immunization. A hard time getting good graft.
That's a more nuanced thing. But we're nervous about transplanting patients with gain of function genetic changes because we give you chemotherapy, clean the field, and then put in the donor cells, but we don't go to 0-0, right? And there's a number of stories of the gain of function clones beating out the donor clones, and you end up back where you started. So it's a nervous thing. So to finish up, I hope I've convinced you, took you on a journey, maybe a little different for this audience, a sort of translational clinical journey to recognize a new human disease caused by heterozygous gain of function variants in STAT6. I think we're scratching the surface of some of the mechanisms, emphasizing sort of increased activity of that axis, but it's still kind of unsatisfying, I think, from a biochemistry perspective of what's happening with that transcription factor doing its job, interacting with DNA. We don't have good information there. And we're happy to have identified some therapies that work well. The other thing that's sort of gratifying in this field is that other people find the same thing because it makes us feel we were right. And so there's been sort of a little bit of explosion of descriptions of this condition, and also showing that the JAK inhibitor treatment, which none of our patients have been on, is also effective because they could access them. So this is our story. We published this now a year or so ago. 2 years, I guess, ago. We had many authors, and we were grateful for them and also grateful for the funding. So I'll end by just saying, they're out there, these experiments of nature, these patients, and I sort of, my passion is that we can build bridges that link our excellent healthcare system, where we funnel the sickest patients to only a relatively small number of hospitals. So although our population's not huge, they end up in one spot, and that's kind of special, I think, with the great basic science firepower that exists on this campus. So anyway, I hope we can do more of this. So thank you.
Stuart, thank you very much. That was a super talk, and you shouldn't apologize for being more clinical translational. I think we need more of that. And this is a Center for Blood Research, after all, and that encompasses everything. So really top talk. So you've had a lot of questions already.
Yeah, well,
a bit of a grilling.
Well, I like
comprehensive exam. Yeah,
it means people are awake. Maybe.
So some more questions.
Hey, Kelly.
Yeah, fantastic, Stuart. That was really great. Hey, so just curiosity, do most of the genes you tend to find fall into like transcription factor and major signal transduction? You don't tend to find ones where you've got kind of gain of function cytokines where they bind really tightly to receptor, or?
Yeah, that's interesting. I mean, we start with, sick patient, we sequence the genome, we filter bioinformatically, and candidates pop out. I would say that a big theme for our lab has been all these transcription factors, and obviously that's not surprising. So we have this one, NFAT1. We found a patient with the NFAT1. This gene MYB is the one that we're working on in the lab. So if anyone's interested in MYB, that's a blood gene. This thing was going to happen. Yes. 155. It tells me I should sit down. TH-PARC, the transcription factor, we just had a paper accepted of the first patient with TH-PARC deficiency who has no CD4 cells. Anyway, so maybe it's not surprising, right, that transcription factors as the master regulators really seem to be the things that we've been finding, but we don't start looking for transcription factors. We start with patients. And I don't think we've found, like you asked about cytokines, like I think we've found lots of changes in the receptors that make them. And immunology used to be, clinical immunology used to be about deficiencies. You know, you didn't have that receptor and then you got infections usually, right? And now kind of the era is you have gain of function and you have autoimmunity, malignancy, allergy. So that's, I would say, the discovery space we're in now. And it's harder to work out a little bit because you're only, usually it's just one copy. So deficiency, right, where you miss both, is like a knockout mouse. It's kind of easier to be sure about.
Well, and I was going to ask you about that too, because I was always struck by those COVID stories where you've got inborn errors and interferons.
Yep.
But then that led to the discovery of people walking around with autoantibodies to interferons. And I wondered if you've seen anything like that, you know, autoantibodies to IL-4 or 13 and things like that.
Yeah, so that's, exactly. So that's the flip side of finding those inborn errors of immunity, genetically encoded, is this autoimmune phenocopy where we make antibodies against interferons and then we, you know, die of COVID and things like that. We haven't found those, like we haven't really looked that much. Typically that's more in the adults. It's usually kind of older. Interestingly, there's a whole kind of ethnic, like it's usually Southeast Asians who have this. There's a whole kind of strong ethnic signal. So on the pediatric side, we don't look that hard for those autoantibodies, but it's usually an adult problem.
That was great. Super interesting. So with the STAT6, I was wondering if this, you showed us the allele frequencies, whether this is like a hotspot for mutations, like a way to tune the immune system, like up or down a little bit, and maybe have small advantages in different situations, different pathogen challenges, whatever.
I think so. And I think that's right. These patients are the ones who are, you know, that thermostat is on to 100 all the time. I think where I'm quite interested in is that next slice, which would be not nearly as bad, but might be, you know, really amenable to these treatments if we can find them. And so in JAK1 gain of function, which is sort of a quite similar condition, that we found some of the all the way on. And now we're recognizing that patients with eosinophilic disease and more autoimmunity are enriched for these. And it also, these conditions then, you know, help us think about new treatment targets. So STAT6 inhibitors are good treatment targets probably for like common forms of allergic disease. It's just a bit harder to work out it's easy to find the outliers a little bit. The experiments are so black and white, it's a little easier to do.
So what was that monoclonal being used for?
Dupilumab for allergies. Yeah.
Just general.
Yeah. So bad atopic dermatitis, for food, so bad eosinophilic disease. Pretty much, it's the wonder medicine of allergic disease, and it's been kind of rolled out in every form of allergic disease.
Yeah, I was just thinking about the flip side, whether people who had like a slight gain of function might have an advantage in place where like worm infections are endemic.
Yeah, I mean, I think that's the population genetic side, right? Is we evolved in a different world. We as a species evolved in a different world to which we live, right? So these beneficial genetic changes aren't maybe so beneficial today. Yeah, no one's really looked at that one yet. I would say the population genetic side.
Thanks for a great talk. So this is linked to the question about autoreactive antibodies. I'm wondering, in these inborn errors of immunity, is it a challenge for treatment with monoclonal antibodies? Is it more common to get reactive antibodies against the treatment? Do you ever see that?
I would say not so much. Yeah. I mean, we'll use the medicines. I would say it's nuanced about the genetics of the underlying condition. But yeah, I don't think I know of any sort of specific, like, wow, we shouldn't treat those guys with monoclonal antibodies because, they'll just make neutralizing antibodies will be a waste of time. I haven't seen that in the literature.
But most of the monoclonals, they're humanized monos, right? Yeah.
Yeah,
So
they're heavily humanized. Yeah, but they're still a bit different. And so people can, but we do still struggle sometimes with these neutralizing antibodies and having to try and get rid of them.
Are there any camel antibodies being therapeutically used?
I don't know. Sorry.
Yeah.
No, I don't know. I'm not so up on that side.
One of the eggs had
a little different than others. The person that had the aneurysm. Now, do we know what the underlying basis was? That they were not a stat 6, or do we know?
Yeah,
that?
he was stat 6. Yeah, so I saw. Yeah.
So aneurysm, that was over and above.
No.
Or was that caused?
Yeah. So I showed you that picture of the JAK-STAT pathways. So these STAT diseases we know. So stat 3, there's stat 1 gain of function, now stat 6. Actually, the aneurysms are part of those conditions, and it seems, maybe it's obvious to you guys, less obvious to me, that genes do different things during embryonic development than they do, sort of in the postnatal life. And so those STAT molecules are really important for vascular development and for the arteries. And so a number of the stat 6 gain of function patients have now had MRIs, and we haven't found more aneurysms, but we've found they're enriched with these unusual variants in the circle of Willis, so that whole circle, and there are anatomical variants that are rare, but these patients keep having them. So I think it's a developmental biology problem with the stat molecules that the vasculature doesn't develop properly.
The circle of Willis says was about 0.25% population has an aneurysm there.
Yeah. I mean, now you're pushing me into.
In addition, they will develop. There could be a stat or any reason.
The stat, yeah. So I guess what I'm trying to say is, we know there's standard anatomy, and then there's variations. Yeah. And when we look at, so again, it's sort of anecdotal at this stage. It's about 10 or 15 of these patients. I don't think there's been any stat 6 gain of function patient who's been scanned that has a normal circle of Willis, like the standard one. So that again just feels like, and there's many patients with that Job syndrome and other things who have had aneurysms and died. So yeah, I'm sorry I left that dangling, but that's part of the disease, as I think is the B-cell lymphoma risk, probably.
But could you use the prevention of future aneurysms?
I wonder that. If we turned off the excess signaling, if we used the JAK inhibitor, would it protect them? They have an abnormality, but they go and develop the aneurysm later. Yeah, I don't know. There's too few to answer.
Try something else in the meantime, but it could be.
Maybe. Yeah, for sure. I think.
I know that anti-macrophages are on and have vascular.
Yes. Yep. Yeah, I think. Yeah, so for me, they have one genetic change that should explain all the problems. I don't like lightning striking twice. And then I think that if we could normalize that exaggerated signaling early in life, we could, minimize. That'd be my hypothesis.
So stat 6 knockout mouse must exist.
They don't get asthma.
The circle of Willis is screwed up?
No, it's fine because it's the turned on too much problem. It's messed up. And I think the other stats can compensate for the developmental biology part. So I think it's too much of a good thing.
Last question.
AP.
Did you, what is that? Was that you? No.
So if the price of sequencing is coming down to about $100, at what point is everybody going to get sequenced at Children's?
Well, yeah, I mean, the companies would say everyone should be, right? It's the problem. So I think it becomes a commodity at this price, and our families will go sequence their kids, right? Even if the healthcare system doesn't do it. The problem is that now the expensive bits are actually like the counseling before we do the test. We might find bad things. We might find that you have, a BRCA genetic change that is, might be very relevant, but in a long time, but this affects your child. So the genetic counseling before the test is now more expensive, I would say, than the test itself. And then there's the bioinformatics back end. So that $100 is like reagent costs. It's not the expensive machine and the reporting, but when you move from tens of millions to 100, that's a profound change.
I was at the Beijing Genome Institute in Shenzhen last year.
Yeah.
And they had one of these gene sequencing robots there. So it's 50,000 whole genome sequences a year, 20 terabases a day.
Yeah.
And they had like 8 of these there. And that was the 4th generation, the 5th generation machines, or 6th generation machines, these little boxes sitting over on the side there. It's just incredible.
Yeah, but we like the medical system is not prepared for this.
No.
Because no one knows.
I mean, the Excel spreadsheet's horrible.
Yeah, AI will help us maybe with this. This is great.
Last call for questions. Look, thanks very much, Stuart. It's fantastic.